Namaste, my dear students, teachers, and researchers of Rumen Microbiology, which is an important sub-discipline of animal nutrition of herbivorous animals. Here are a series of presentations of microbes present in the gastrointestinal tract of herbivorous animals. The author and the presenter is Professor Devkinandan Kamra, who retired as ICAR National Professor from Animal Nutrition Division of Indian Veterinary Research Institute, Izzatnagar, India. The lectures will be useful and informative for students and young researchers of animal nutrition and allied subjects. The chemical composition shows that there are lignin, cellulose, hemicellulose, starch, and sugar-containing feeds. Some of them might have toxic materials like silica, tannins, saponin, alkalides, oxalates, nitrates, and sulfates, etc. The animals themselves cannot degrade these feeds as they have no enzyme of animal origin required for the degradation of these toxic fields. Therefore, they depend upon the microbes present in some part of the gastrointestinal tract, the fermentation sacs, which help the animal in getting energy out of these feeds. And the major sacs, the fermentation sacs, are rumen, pseudorumen, cecum, and colon. Herbivorous animals are classified into pre-gastric and post-gastric fermenters. The ruminants like cattle, buffalo, sheep, goat, and wild ruminants have 4-chambered stomach. The pseudoruminants have a 3-chambered stomach. They do almost the same functions as the ruminants do. The post-gastric fermenters can be classified into 3 groups: colon fermenters, cecum fermenters, and mega-herbivorous, that is monogastric animals. So the cecum fermenters have a gastrointestinal tract about 10 to 13 times the length of their body to accommodate more roughage feed and increased retention time for such feeds. The similar hind-gut fermentation like rabbit, hare, and pikas formed in the cecum are passed through the large intestine and subsequently re-ingested to allow another opportunity to absorb nutrients. The cecotrophs are surrounded by a layer of mucus which protects them from acids in the stomach and which doesn't inhibit nutrient absorption in the small intestine. The coprophagy is the process practiced by some rodents such as capybara, guinea pig, hare, and rabbits. This process is also beneficial in allowing the restoration of the microflora population or the gut flora. These microbes are found in the digestive organ of living creatures and can act as a protective against that strengthens the immune system. As far as the feed degradation is concerned, the foregut fermentation is generally considered more efficient. The monogastric animals cannot digest cellulose as efficiently as the ruminants. Hindgut fermenters allows animals to consume small amount of low quality forage all day long and thus survive in the conditions where the ruminants might not be able to obtain nutrients adequate for their needs. The ability to process food more rapidly than foregut fermenters gives hind-cut fermenters an advantage at the large body size, as they also are able to accumulate significantly larger food intake. Now the question arises, what the microbes do in the rumen. They convert feeds from non-utilizable form, that is lignocellulosic feeds, as the animals have no enzyme excreted which can digest these feeds, to utilizable form of energy, that is volatile fatty acids, the microbial protein, etc., and provide better quality protein to the animal. They help in conversion of a non-edible products, cereal straws, into good quality foods and fiber like milk, meat, and wool. When the microbes are so important for feed digestion in the rumen, why is rumen microbiology a neglected area in many countries? There is no well-established culture collection or depository to keep the isolated and standard cultures. Relatively difficult to cultivate and maintain rumen microbes, as these microbes require very specific and sophisticated laboratory facilities. Low priority area for microbiologists, biotechnologists, and molecular biologists. Researchers prefer to work on clinical, industrial, and food microbiology. There is a lack of coordination between nutritionists and microbiologists, and now a coordinated research project has been started specifically for rumen microbes by the Indian Council of Agricultural Research, New Delhi, India. The rumen environment, osmotic pressure is very high in the rumen, tune of 260 to 280 millios per kg rumen liquor before feeding. After offering feed, it rises by 50%. The osmotic pressure more than 400 millios per kg is detrimental for cellulose degradation. The high pressure is only for the short duration, just after feeding, so that there is no adverse effect on cellulose degradation. The rumen microbes can sustain this high osmotic pressure, therefore they are classified as moderately halophiles. The second important characteristics of the rumen environment is buffering capacity. The rumen content is buffered at pH ranging from 6.5 to 6.9, that is almost neutral. Postprandial changes might vary from 5.5 to 7.3 depending upon the diet. If we are feeding high energy diet, pH might go as low as 5.5, and if the feed contains high roughage diet, pH might go up to 7.3. Carbonates, phosphates, volatile fatty acids determine the buffering capacity of the rumen content. It is an important rumen characteristic affecting the consortium of rumen microbes under a set of feeding regimen and chemical composition of feed. General function of rumen microbes: they are bioconversion of feed, fibrolysis, which animals cannot do by themselves, proteolysis, most of the cellulose degrading bacteria are protease negative, therefore they depend upon other microbes to make the substrate free of protein for cellulose degradation. Of course, lipolysis is present in the rumen content, but not very important because the lipid content in the feed is very low. Microbial biomass synthesis, any type of protein, even the non-protein nitrogen sources, can be converted to good quality feed. Microbial protein, which is used by the animals as a source of nitrogen. Specific function of microbes are: the bacteria degrade most of the toxic materials like saponins, tannins, alkaloids, nitrates, sulfate, and oxalates, etc. The archaea are the hydrogen utilizers, methane producers. They are highly combustible gas, that's hydrogen is converted to methane, less dangerous gas than hydrogen. The protozoa, it is a pH stabilizer, engulfs all the sugars and easily fermentable carbohydrates included in the feed and protect them from immediate fermentation. Otherwise, the organic acid so produced will lower the pH and lead to acidosis of the rumen. When the protozoa are present, the sugar-based feeds are protected and sugars are released throughout the day and maintain a constant source of energy to the animals. Fungi. They act as a biological knife and cut a large particle of feed into several hundred small particles which have larger surface area than those on one large particle, on which more bacteria can attach, and smaller particles are easily engulfed by the protozoa. Thus, the fermentation is more complete. In the absence of fungi from the rumen, the degradation of lignocellulosic feeds are adversely affected. The last group is the bacteriophages. These are bacterial viruses, and bacteria is poorly digested in the rumen. The bacteriophages help in bacteria disintegration and release of amino acid for the animals' use. Infection of calf rumen with microbes. Mouth-to-mouth contact, feeding and grazing together, aerial and dust contamination may cause infection of bacteria and fungi from adult animals to the younger ones. But in the case of protozoa, mouth-to-mouth contact, feeding and grazing together might be the reason for infection of protozoa into the rumen. Distribution of hydrolytic enzymes in different fractions of rumen content. About 80 to 90% of total hydrolytic enzymes are attached with particulate material, while rest of the 8 to 10% activity is present in the cellular or extracellular fractions. Therefore, it is essential that while studying the enzymes and microbes of fibrous feed given to animals, the sampling of the rumen liquor or rumen content should be taken from the particulate material of rumen contents. The chemical composition of feeds, as I told earlier, shows that it is having cellulose, hemicellulose, cell contents are easily fermentable components, but lignin, tannin, ash, and other identified or unidentified toxic materials are difficult to ferment. The little group of compounds even inhibit the degradation of easily fermentable carbohydrates or components. The establishment of rumen microbes. The bacteria on day 1 in newly born calf intestine is sterile and free of any microbes. Within the first week, aerobic and anaerobic bacteria are very high, that is 10 to the power 8 to 10 to the power 9 per gram of rumen content as a contamination from soil. In second week, the number is reduced to half. The anaerobic bacteria start growing and the true rumen bacteria start establishing after second week of age of the animal. Normally, the rumen bacteria in the first 5 to 8 hours and up to 4 days E. coli, Clostridium welchi, and Streptococcus bovis, they get established into the rumen. After 3 weeks, lactobacilli, 5 weeks, lactic acid utilizing bacteria, 6 weeks, amylolytic bacteria and Prevotella, 6 to 10 weeks, cellulose degraders, Methanogens, Vibrio, Ruminococcus, and Fibrobacter get established in the rumen. From week 1 to 12, the proteolytic bacteria get starting establishing in the rumen content. From week 3 to 9, the protozoa get established, and from week 9 to 13, the normal population of microbes starts establishing in the rumen. The protozoa start establishing within 2 weeks of age of the animals. The starch degrading enterodermium gets established first of all, followed by cellulose degrading protozoa like Diplodinium, Eudiplodinium, Polyplastron, Epidinium, and the last of all, sugar fermenting protozoa, the holotrig, get established in 40 to 50 days of age of the animal. The fungus gets established in 2 weeks of time, and the level up to that of adult animal reaches in 6 to 8 weeks. The Rumen microbial ecosystem consists of 5 different microbial groups. Bacteria, they act on the surface, create cavities, hydrolyze substrate, and change it into monomers and fermentation and products. The bacteria act on the surface, create cavities, hydrolyze substrate, and change it into monomers and fermentation and products. The fungus penetrates the substrate, breaks down the larger feed particle into smaller pieces to increase the surface area for bacterial attachment and engulfment by the protozoa for further fermentation. Protozoa, they feed upon small particles of feed, hydrolyze substrates, and excrete monogastric sugars for further rumen fermentation of substrate. The bacteriophages, they help in turnover of bacterial protein and make them available to the animal. And the last group is the archaea. Mainly they are methanogens. They help in continuity of sugar fermentation, acts as a hydrogen sink and converts highly combustible hydrogen to less dangerous methane gas, as methane has lower octane value than hydrogen. There is a large difference in the number of bacteria and protozoa, but they each can account for 40 to 50% of the microbial biomass. It might be because of the much larger size of protozoa than bacteria. The bacteria are represented by about 200 species and protozoa by 25 genera. Fungus is responsible for 3 to 5% of total biomass, and the number of genera is 7, and each one of them is cellulase positive. Although their number is small, but they play a very significant role in fiber degradation. The bacteriophages help in degradation of bacteria and make amino acid available to the animal. There are a few constraints in the rumen. First is anaerobiosis. The rumen microbes require a very low redox potential for their growth. It is sometimes minus 350 millivolts of anaerobiosis. Then there is a high osmotic pressure. Because of high osmotic pressure, many of the water living microorganisms, they are not able to survive in the rumen. Then there is very high buffering capacity. So because it can tolerate a large quantity of acids or bases, which can protect the microorganism in the rumen, and they don't allow any external microbe to survive in the rumen. Then there are some antimicrobial compounds which are produced by different microbes in the rumen. They are inhibitory for the growth of other microbes. All these constraints in the rumen are responsible for stability of the rumen microbial ecosystem, because the non-rumen microbes are not able to survive under these conditions. Depending upon the studies conducted so far in the rumen, especially in the last 50 years, a lot of work on the rumen microbiology has been conducted, but majority of the microbes are still unculturable, as we are not aware of their requirement for in vitro cultivation. Very few studies on metagenomics to understand the rumen microbial ecosystem have been conducted. More studies on this technique need to be undertaken to better understand the rumen microbial ecosystem. Exact microbial and enzyme profile are not known for degradation of a particular fiber-based diet. On each of the fiber, the enzymes and the microbial profile might be different. Therefore, selective stimulation in the rumen, selective removal of the non-useful microbes from the rumen might make the job easier to understand the rumen fermentation and to manipulate it for better animal productivity on high roughage fed animals. Thank you very much.